This video contains audio content only. There are no visual elements or images accompanying the audio. If you're expecting to see video footage, please note that this is an intentional audio-only presentation. We recommend listening to this content rather than watching the screen. Thank you for your understanding and enjoy the audio. Join us as we unravel the captivating story of blood, a journey spanning billions of years of evolution. In simple terms, we'll explore how this vital fluid transformed from basic diffusion in single-celled organisms to the complex circulatory systems we see today. We'll delve into the origins of blood cells, the rise of red blood cells and oxygen transport, and the surprising role of infectious diseases in shaping blood type diversity. Get ready to discover the remarkable evolutionary history hidden within your veins. Chapter 1. From primordial soup to circulatory flow. Imagine our planet billions of years ago. The oceans, a vast and murky primordial soup, teemed with life's simplest forms. Single-celled organisms, microscopic pioneers, floated freely, their existence a delicate dance with their surroundings. They absorbed nutrients and expelled waste directly through their cell membranes, a process called diffusion, sufficient for their minuscule size. Life, in this early stage, was a slow, intimate exchange with the environment. But evolution, the tireless tinkerer, had grander designs. Life began to experiment with multicellularity, single cells joining forces to create larger, more complex organisms. This was a monumental leap, but it presented a new, formidable challenge. As organisms grew larger, the distances within their bodies increased dramatically. The once efficient diffusion could no longer keep pace. Imagine a bustling city relying solely on foot traffic for transportation. As the city expands, getting food and supplies to its outermost reaches, or removing waste, becomes increasingly difficult. Similarly, these early multicellular organisms faced a logistical nightmare: how to effectively transport vital materials across ever-growing internal distances. Some of the earliest multicellular animals, like sponges, tackled this problem with a relatively simple solution. They capitalized on their aquatic environment, creating channels and pores throughout their bodies that allowed seawater to circulate freely within them. This continuous flow of seawater brought nutrients and oxygen directly to their cells, while simultaneously carrying away waste products. These organisms, called diploblasts, representing an early branch on the animal tree of life, demonstrated a clever adaptation, but this system had its limits. It relied heavily on the surrounding water and offered little control over the direction or speed of circulation. A more sophisticated solution emerged with the arrival of triploblasts, a group of animals that includes the vast majority of animal species today. These organisms, characterized by 3 germ layers during embryonic development, pioneered the use of internal, extracellular circulatory fluids. This was a game-changer. Instead of relying on the external environment, triploblasts created their own internal transport systems, effectively shrinking the functional diffusion distance. Imagine the city now developing a network of roads and delivery vehicles. Goods can now reach distant locations quickly and efficiently. Similarly, the internal circulatory fluids of triploblasts provided a dedicated highway for nutrients, oxygen, and waste products, enabling these organisms to grow larger and more complex. These early circulatory fluids were propelled through the body in a variety of ways. Some organisms, like many invertebrates, employed cilia, tiny hair-like structures on the surfaces of cells, to create gentle currents that moved the fluid. Others, particularly larger animals, developed muscle-powered systems. Muscles surrounding fluid-filled cavities contracted in coordinated waves, pushing the fluid through the body in a process called peristalsis. Think of squeezing a tube of toothpaste. The pressure you apply moves the toothpaste in one direction. This same principle, applied within the body, provided a more powerful and controlled method of fluid transport. Crucially, these early circulatory systems also began to incorporate mechanisms for directional flow. Coordinated peristaltic waves, like those seen in the digestive tracts of many animals, ensured that the fluid moved in a consistent direction. The evolution of one-way valves, flap-like structures that prevented backflow, further refined this control, ensuring efficient delivery and removal of substances. The relationship between body size and circulatory system complexity is a testament to the power of evolutionary adaptation. As organisms increased in size, their surface area to volume ratio decreased. This meant that the surface area available for diffusion, relative to the volume of the organism, became smaller and smaller. Imagine a balloon inflating. As it gets bigger, the surface area grows, but the volume grows even faster. This decreasing surface area to volume ratio posed a significant problem for larger organisms relying on diffusion. Circulatory systems provided a crucial workaround, enabling organisms to overcome the limitations of diffusion and achieve greater size. Consider flatworms, relatively simple triploblasts that lack a coelom, a fluid-filled body cavity. Their flattened body shape maximizes their surface area, allowing for some reliance on diffusion. However, their circulatory system, while present, is less complex than that of animals with a coelom. The coelom, a defining feature of many animals, plays a significant role in circulation. It's a cavity lined with a special tissue called mesothelium, filled with a fluid called coelomic fluid. This fluid, often containing specialized cells called coelomocytes, acts as a circulatory medium, transporting materials throughout the body. In many animals, the coelom works in concert with a blood vascular system, a network of vessels that carry blood. These two systems, working together, provide a highly efficient and adaptable circulatory network, enabling organisms to achieve even greater size and complexity. The evolution of the coelom and the increasingly sophisticated blood vascular system set the stage for another critical development: the emergence of specialized cells within the circulatory fluid. These cells, with their unique functions, would further refine the transport and defense capabilities of circulatory systems, paving the way for the evolution of blood as we know it. The story of these specialized cells, the forerunners of modern blood cells, is the subject of our next chapter. Chapter 2. The Cellular Saga. Tracing the Ancestry of Blood Cells. The previous chapter unveiled the ingenious ways circulatory systems evolved, transporting vital substances throughout increasingly complex bodies. Now, we delve into the very fluid that courses through these circulatory highways, blood, and trace the fascinating ancestry of its cellular constituents. Our exploration begins, surprisingly, not within the circulatory system itself, but in the ancient oceans, with life's simplest forms, unicellular organisms. These single-celled pioneers, though lacking the intricate circulatory networks of their multicellular descendants, already possessed the fundamental machinery that would eventually give rise to our complex immune system. A prime example is Capsospora ow zarzaki, a unicellular organism more closely related to animals than to plants or fungi. Capsospora, in its amoeboid form, bears a striking resemblance to macrophages, a type of white blood cell that plays a crucial role in our immune defenses. This resemblance goes beyond a superficial similarity. It extends to the very core of their cellular machinery. Both Capsospora and macrophages are voracious phagocytes, meaning they engulf and digest foreign particles, pathogens, and cellular debris. Imagine these cells as tiny vacuum cleaners, patrolling the body and keeping it clean. This shared phagocytic ability is a compelling clue to a deep evolutionary connection. Further strengthening this link is the remarkable similarity in their transcriptional profiles, the genetic blueprints that dictate cellular function. Out of 325 genes highly expressed in Capsospora and 545 in macrophages, a significant overlap of 101 genes is dedicated to phagocytosis and lysosome function. Lysosomes are the cell's recycling centers, breaking down waste materials and preparing them for reuse or expulsion. The shared genetic toolkit between Capsospora and macrophages suggests that the core machinery for phagocytosis was established long before the evolution of multicellular animals. This ancient heritage is further underscored by Capsospora's possession of philopodia, thin, finger-like projections that extend from the cell surface. These philopodia, used for sensing and manipulating the environment, are reminiscent of the structures used by macrophages to reach out and capture their targets. Even the internal structure of Capsospora, with its numerous vacuoles or storage compartments, mirrors the cytology of macrophages. Intriguingly, the similarities extend beyond macrophages. Capsospora also shares transcriptional similarities with other mammalian cells known for their phagocytic potential, such as hepatocytes, liver cells, fibroblasts, connective tissue cells, and adipocytes, fat cells. These cells, though not part of the immune system, can, under certain conditions, engulf and digest particles, and they share lysosome-related genes with Capsospora. This broader similarity hints at a more widespread ancestral role for phagocytosis and cellular function. Gene ontology analysis, a method for studying the functions of genes, further supports this notion. It reveals that the 325 genes highly expressed in Capsospora are significantly enriched for functions related to phagocytosis and lysosomes, compared to the 2,252 genes expressed at lower levels. Moving along the evolutionary timeline, we encounter the first multicellular animals, sponges. These seemingly simple creatures possess specialized cells called archaeocytes, which, like macrophages, exhibit phagocytic activity. Sponges, representing the oldest extant animal lineage, offer a glimpse into the early stages of immune cell evolution. Next, we encounter tunicates, marine invertebrates more closely related to vertebrates than are sponges. This phylogenetic proximity makes tunicates particularly relevant in our quest to trace the ancestry of blood cells. Tunicates, like sponges and vertebrates, possess phagocytes that share transcriptional similarities with vertebrate macrophages. Comparing gene expression profiles across mouse, tunicate, sponge, and Capsospora reveals a surprising pattern. Despite their vast evolutionary distance, mouse macrophages show higher transcriptional similarity to Capsospora than to many other mouse cell types. This unexpected kinship reinforces the notion of an ancient, conserved phagocytic program. To ensure these findings were not artifacts of the data analysis, researchers employed multiple datasets generated using different techniques, RNA-seq and CAGE, confirming the robustness of the macrophage-Capsospora connection. The emergence of phagocytes, these ancient sentinels of the immune system, marks a crucial step in the evolution of blood cells. But the story doesn't end there. How did the diverse array of blood cell types we see today arise from these ancestral phagocytes? The answer lies in the intricate dance of genes and their regulation. The myeloid lineage, the branch of blood cell development that gives rise to phagocytes, appears to represent a default pathway. This means that, in the absence of other instructions, blood cell precursors tend to develop into phagocytes. The diversification of blood cells, the emergence of non-phagocytic lineages involved in functions like oxygen transport, required a shift away from this default pathway. A key player in this shift is a transcription factor called CEBP-alpha. Transcription factors are proteins that control the expression of other genes, acting as molecular switches that turn genes on or off. CEBP-alpha is essential for the development of myeloid cells, including macrophages. Experiments have shown that overexpressing CEBP-alpha in non-phagocytic blood cell precursors can induce their transdifferentiation into macrophages. Conversely, repressing CEBP-alpha can divert cells away from the myeloid lineage. This repression is achieved, in part, by polycomb genes, which act as molecular brakes, silencing the expression of specific genes. Ablating, or removing, polycomb genes in non-phagocytic blood cells leads to the derepression of CEBP-alpha and the subsequent conversion of these cells into phagocytes. These experimental manipulations, though performed in modern cells, offer a glimpse into the evolutionary processes that shaped blood cell diversity. The interplay between CEBP-alpha and polycomb genes likely played a crucial role in the emergence of non-phagocytic blood cell lineages. By repressing CEBP-alpha in a subset of ancestral phagocytes, polycomb genes paved the way for the evolution of alternative cell fates. This initial diversification was further refined by the evolution of lineage-specific transcription factors, such as PAX5 for B cells and GATA1 for erythroid, megakaryocytic cells, which orchestrated the development of specialized blood cell types with distinct functions. These later evolving transcription factors, building upon the foundation laid by the CEBP-alpha polycomb interplay, would eventually give rise to the complex blood system we see in vertebrates today. The next chapter will explore the evolution of one of these specialized cell types, the red blood cell, and its critical role in oxygen transport. Chapter 3. A Symphony of Systems, the Rise of Vascular Networks. Life's relentless drive for greater complexity and efficiency has sculpted many remarkable adaptations, but few are as elegant and essential as the circulatory system. From the humble beginnings of simple diffusion to the intricate symphony of vessels, hearts, and specialized circulatory pathways, the evolution of these systems is a testament to nature's ingenuity. In the primordial oceans, single-celled organisms thrived, their microscopic world a realm where diffusion reigned supreme. Nutrients and waste products flowed freely across their cell membranes, a direct exchange with their surroundings. But as life ventured into the realm of multicellularity, this intimate connection with the environment became a liability. Larger organisms, with their increased internal distances, faced the challenge of efficiently transporting vital substances across their bulk. Diffusion, once a lifeline, became a bottleneck. The limitations of diffusion stem from the fundamental relationship between surface area and volume. As an organism grows larger, its volume increases much faster than its surface area. Imagine a simple cube. If you double its length, its surface area increases fourfold, but its volume increases eightfold. This means that the surface area available for diffusion, relative to the volume that needs servicing, shrinks dramatically. For larger organisms, diffusion alone simply cannot keep pace with the demands of metabolism and waste removal. Early multicellular animals, like flatworms, exemplified the limitations of relying solely on diffusion. These ecoelemates, lacking a body cavity, have a flattened body shape that maximizes their surface area, bringing as many cells as close as possible to the external environment. Their low metabolic demands and reliance on simple diffusion across their thin bodies allowed them to thrive in their specific ecological niches. But this strategy limited their potential for size and complexity. A more sophisticated solution emerged with the evolution of the coelom, a fluid-filled body cavity lined by a specialized tissue called mesothelium. This innovation marked a major transition in animal evolution. The coelom, present in most triploblasts, animals with 3 embryonic germ layers, created an internal environment distinct from the external world. This internal space, filled with coelomic fluid, acted as a circulatory medium, transporting nutrients and waste products throughout the body. In ecoelemates like flatworms, the space between the ectoderm, outer layer, and endoderm, inner layer, is filled with a meshwork of cells called parenchyma. This parenchyma, while providing structural support, limits the potential for efficient circulation. In contrast, the coelom, with its fluid-filled space and mesothelial lining, facilitates more efficient transport. The mesothelium, a thin layer of epithelial cells, regulates the movement of substances into and out of the coelomic fluid. Specialized cells within the coelomic fluid, called coelomocytes, can also play a role in nutrient transport, waste removal, and even immune defense. Animals like some small, aquatic invertebrates rely primarily on coelomic circulation. Fluid movement within the coelom is achieved through the coordinated beating of cilia on the mesothelial cells or through muscle contractions that create peristaltic waves, similar to the squeezing motion that propels food through our digestive tract. However, coelomic circulation, while an improvement over simple diffusion, lacks the efficiency and targeted delivery of more complex systems. The transition from coelomic circulation to blood vascular systems marked another major evolutionary leap. This transition involved the emergence of specialized vessels, sinuses, and pumping organs. Vessels, tubes of varying sizes, provided dedicated conduits for circulatory fluid, now called blood. Sinuses, open spaces within tissues, allowed for direct exchange between blood and surrounding cells. Pumping organs, the precursors of hearts, provided the driving force for blood circulation. Invertebrates showcase a diverse array of blood vascular systems. Open circulatory systems, found in animals like arthropods, insects, spiders, crustaceans, and some mollusks, snails, clams, allow blood to directly bathe tissues. Imagine a sprinkler system irrigating a garden. The water spreads over a wide area, but the delivery is not precise. Similarly, in open circulatory systems, blood flows freely through body cavities, directly contacting cells and delivering nutrients and oxygen. While efficient for smaller organisms, open systems lack the targeted delivery and pressure control needed for larger, more active animals. Closed circulatory systems, characteristic of vertebrates, animals with backbones, confine blood within vessels. Think of a network of pipes delivering water to individual houses. The flow is controlled and directed, ensuring efficient delivery to each location. Similarly, in closed systems, blood circulates within a network of arteries, veins, and capillaries, allowing for precise control of blood flow and efficient delivery of oxygen and nutrients to specific tissues. This targeted delivery is crucial for supporting higher metabolic rates and activity levels. The evolution of hearts, specialized pumping organs, further enhanced the efficiency of closed circulatory systems. From the simple, two-chambered hearts of fish, which pump deoxygenated blood to the gills and then to the body, to the three-chambered hearts of amphibians and most reptiles, which allow for some mixing of oxygenated and deoxygenated blood, to the four-chambered hearts of birds and mammals, which completely separate oxygenated and deoxygenated blood, the evolution of hearts mirrors the increasing demands of vertebrate lifestyles. The four-chambered heart, with its separate circuits for pulmonary, lung, and systemic, body, circulation, maximizes oxygen delivery and supports the high metabolic rates of birds and mammals. The evolution of increasingly complex circulatory systems was driven by relentless selective pressures. Larger body size, increased activity levels, and the colonization of new environments demanded more efficient transport of oxygen and nutrients. Closed circulatory systems, with their dedicated vessels, hearts, and blood pressure regulation, provided the necessary infrastructure for these adaptations. The evolution of blood pressure regulation, the ability to control the force of blood flow, further refined circulatory function, ensuring efficient delivery of oxygen and nutrients to even the most distant tissues. The interplay between circulatory system evolution and metabolic rate is particularly striking. Closed circulatory systems, with their efficient oxygen delivery, enabled animals to achieve higher metabolic rates, which in turn allowed for greater activity levels and the exploitation of new ecological niches. The evolution of warm-bloodedness, endothermy, in birds and mammals is a prime example of this interplay. Endothermy, the ability to maintain a constant body temperature independent of the environment, requires a high metabolic rate, which is supported by the efficient oxygen delivery of a four-chambered heart and a closed circulatory system. The story of circulatory system evolution is a tale of increasing complexity and refinement, driven by the constant pressure to adapt and survive. From the simple diffusion of single-celled organisms to the intricate circulatory pathways of vertebrates, the evolution of these systems has shaped the diversity of animal life and enabled the colonization of virtually every habitat on Earth. These circulatory networks, the very lifeblood of complex organisms, stand as a testament to the power of evolutionary innovation. Chapter 4. Crimson Rivers. The evolution of red blood cells and oxygen transport. The previous chapter explored the rise of circulatory systems, nature's ingenious solution to the transport challenges faced by increasingly complex organisms. Now, we delve into the very fluid that pulses through these living pipelines, blood, and specifically, the remarkable evolutionary journey of red blood cells and the oxygen-carrying protein that gives them their crimson hue, hemoglobin. The emergence of red blood cells and hemoglobin revolutionized oxygen transport, providing a dramatic leap in efficiency that fueled the rise of larger, more metabolically active organisms. Before this innovation, oxygen transport relied heavily on simple diffusion, the passive movement of molecules from areas of high concentration to areas of low concentration. The story of hemoglobin begins with a family of proteins called globins. These proteins, characterized by a unique three-dimensional structure known as the globin fold, possess a remarkable ability to bind to metal ions. This binding capacity, combined with the precise architecture of the globin fold, allows globins to interact with oxygen. The key to this interaction lies in a small, ring-shaped molecule called heme, which contains an iron atom at its center. This iron atom acts as a molecular docking station for oxygen, allowing hemoglobin to capture oxygen in the lungs or gills and then release it to tissues throughout the body. Hemoglobin, the oxygen-carrying workhorse of our blood, is just one member of the diverse globin family. While its primary role in vertebrates is oxygen transport, globins have evolved to perform a surprising variety of functions in different animal lineages. In some clams, for instance, hemoglobin acts as an oxygen reservoir, storing oxygen for use by nerve cells when oxygen levels in the surrounding water are low. Certain nematodes, tiny roundworms, utilize hemoglobin in the lining of their mouths, ensuring a steady supply of oxygen to their feeding apparatus even in oxygen-depleted environments. Even the humble bacterium E. coli possesses a type of globin, though its role appears to be sensing oxygen levels rather than transporting it. This remarkable versatility showcases the adaptability of the globin fold and its importance in the evolution of diverse biological processes. The evolutionary history of hemoglobin is a long and winding tale, stretching back hundreds of millions of years. Traces of heme, the iron-containing component of hemoglobin, have been found in fossilized mosquitoes dating back 46 million years, providing a tantalizing glimpse into the ancient history of this protein. But hemoglobin itself predates these fossilized insects by a vast expanse of time. Molecular studies suggest that the ancestral globin genes arose over 700 million years ago, long before the appearance of the first animals. The type of hemoglobin found in vertebrates, with its 4 globin subunits and cooperative oxygen binding, is thought to have evolved around 450 million years ago, coinciding with the emergence of jawed vertebrates. This evolutionary innovation greatly enhanced oxygen transport efficiency, providing the fuel for the rise of larger, more active vertebrates. The 4 subunit structure of vertebrate hemoglobin allows for cooperative binding, meaning that the binding of one oxygen molecule makes it easier for subsequent oxygen molecules to bind. This cooperative behavior ensures efficient oxygen loading in the lungs and efficient unloading in tissues. As vertebrates diversified and colonized new environments, hemoglobin continued to evolve, adapting to the specific challenges of each lineage. In high-altitude environments, where oxygen is scarce, some mammals have evolved hemoglobins with an increased affinity for oxygen, allowing them to extract oxygen more efficiently from the thin air. Diving mammals, such as whales and seals, have evolved hemoglobins that can store larger amounts of oxygen, enabling them to remain submerged for extended periods. These adaptations, sculpted by natural selection, highlight the remarkable plasticity of hemoglobin and its crucial role in vertebrate evolution. The evolution of red blood cells, the specialized cells that carry hemoglobin, is intimately linked to the rise of efficient oxygen transport. These cells, packed with hemoglobin, act as microscopic oxygen tankers, circulating throughout the body and delivering oxygen to tissues. The emergence of red blood cells, along with the evolution of hemoglobin, overcame the limitations of diffusion, paving the way for the evolution of larger, more complex organisms. The connection between oxygen availability and metabolic activity is fundamental to understanding the evolution of complex life. Oxygen is the essential ingredient for aerobic respiration, the process by which cells convert nutrients into energy. Enhanced oxygen transport, facilitated by red blood cells and hemoglobin, allowed for a dramatic increase in metabolic rates, fueling the evolution of more energetic and complex life forms. This increased metabolic capacity enabled animals to sustain higher activity levels, develop more sophisticated nervous systems, and colonize new environments. The rise of red blood cells and efficient oxygen transport was not a single event but a series of evolutionary innovations, each building upon the previous one. From the ancestral globins that first bound to oxygen to the complex, cooperative hemoglobin of vertebrates, the evolution of this protein has been a driving force in the diversification of animal life. The crimson rivers that flow through our veins, carrying oxygen to every cell in our bodies, are a testament to the power of evolutionary adaptation and the enduring legacy of this remarkable protein. Chapter 5. Blood feuds. Evolutionary arms races and the ABO blood group system. The very essence of life, blood, pulsates with a history far more dramatic than simply carrying oxygen. It's a battlefield, a molecular arena where hosts and pathogens clash in a never-ending evolutionary arms race. The familiar A, B, and O blood groups, known to us from blood transfusions and paternity tests, are not mere biological markers. They are scars of this ongoing conflict, testaments to the relentless pressure exerted by infectious diseases. The ABO blood group system, a key player in this evolutionary drama, is defined by the presence or absence of specific molecules, called antigens, on the surface of red blood cells. These antigens, acting like cellular flags, can trigger powerful immune responses if recognized as foreign. This system, far from being unique to humans, has deep evolutionary roots, stretching back millions of years in the primate lineage, long before the human-chimp divergence. This shared heritage underscores the ancient origins of this complex system and its importance in the ongoing struggle against disease. The A, B, and O blood types arise from subtle variations in a single gene that codes for glycosyltransferases, enzymes that attach specific sugar molecules to the surface of red blood cells. These sugar molecules form the A and B antigens. The O blood type, originally termed C, arises from a mutation in this gene that renders the enzyme inactive, resulting in the absence of both A and B antigens. Hence the later designation, O, for ohne, meaning without, in German. The seemingly minor differences between the A and B versions of this gene, often involving just a few amino acid substitutions, have profound consequences for blood type and disease susceptibility. For example, the A and B transferases differ at 4 key amino acid positions: 176, 235, 266, and 268. The amino acids at positions 266 and 268 are particularly critical, dictating which sugar molecule, N-acetyl-galactosamine for A or galactose for B, the enzyme attaches to the red blood cell surface. Researchers have even developed a code table that links specific amino acid combinations at these crucial positions to our B antigen specificity, allowing them to decipher the potential blood type of extinct species or explore the vast diversity of ABO genes across different animal lineages. The evolution of this system is a dynamic process, governed by what scientists call birth and death evolution. This model describes a continuous cycle of gene duplication, mutation, and loss within the alpha-1, 3-gal, NAC, T gene family, which includes the ABO gene. This constant genetic shuffling creates new variants, some of which may confer selective advantages, while others are lost to evolutionary time. This dynamic process, driven by the relentless pressure of infectious diseases, has resulted in the remarkable diversity of blood types observed across the animal kingdom. The persistence of multiple blood types, rather than a single, universal, type, suggests that each variant offers some protection against different pathogens. This diversity creates a population-level resilience, a safeguard against the ever-shifting landscape of infectious disease. Consider the complex relationship between blood type and malaria, a parasitic disease transmitted by mosquitoes. Individuals with type O blood show a remarkable resistance to severe malaria, while those with type A are more vulnerable. This difference in susceptibility stems from the way malaria parasites interact with the antigens on the surface of red blood cells. The parasite Plasmodium falciparum, responsible for the most severe forms of malaria, uses a protein called RIFIN to bind to red blood cells. RIFIN binds more strongly to type A antigens than to type O, making type A individuals more susceptible to severe complications like cerebral malaria, a life-threatening condition where infected red blood cells clog blood vessels in the brain. The protective effect of type O extends beyond malaria. Studies suggest that type O individuals may also be less susceptible to severe cholera and plague, diseases caused by bacterial infections. Conversely, type A individuals appear to be more susceptible to smallpox, a viral disease. These varying susceptibilities highlight the complex interplay between blood type and disease, a relationship shaped by millions of years of co-evolution between hosts and pathogens. Intriguingly, some blood types may have offered advantages in ancestral environments that are now considered disadvantages in the modern world. For example, individuals with type A blood are more prone to blood clots. While this increased clotting tendency can be problematic today, in ancestral environments where injuries and infections were common, it may have been beneficial. Rapid clotting could have prevented excessive bleeding from wounds or helped contain infections, providing a crucial survival advantage in a world without modern medicine. The ABO blood group system is not the only example of an evolutionary arms race playing out in our blood. The evolution of blood clotting itself is a testament to this ongoing struggle. The intricate cascade of proteins involved in clotting, while essential for preventing excessive bleeding, can also lead to dangerous blood clots in modern humans, particularly in the context of cardiovascular disease. However, in ancestral environments, where injuries were common and medical care non-existent, rapid and efficient clotting would have been a lifesaver, preventing fatal blood loss from even minor wounds. This ancient adaptation, honed by countless generations of natural selection, highlights the trade-offs inherent in evolutionary solutions. What was once a crucial survival advantage can become a liability in a changed environment. The diversity of blood types, shaped by the constant pressure of infectious diseases, has likely played a crucial role in human survival. In unpredictable environments, where different pathogens waxed and waned, a diverse array of blood types would have ensured that at least some individuals within a population were resistant to any given disease. This population-level resilience, a product of millions of years of evolutionary fine-tuning, is a powerful testament to the importance of genetic diversity in the face of ever-changing environmental challenges. The story of blood, far from being a simple tale of oxygen transport, is a complex saga of conflict, adaptation, and the enduring power of evolution. From single-celled diffusion to complex circulatory systems and the diverse world of blood cells, evolution's drive for efficiency has shaped life profoundly. The ancient origins of immunity, the dynamic evolution of blood groups, and hemoglobin's adaptability highlight the interconnectedness of life and the constant interplay between adaptation and environmental pressures. These insights into our biological history not only deepen our understanding of life's complexity but also offer potential for advancements in areas like disease treatment and personalized medicine. What wonders will further exploration uncover? Share your thoughts and questions below. Like, subscribe, and suggest topics for future videos. Keep exploring, keep questioning, the journey of discovery never ends.